Thank you so much. Thank you, Adam. That was such a nice introduction. All right, so I'm going to just jump right in and talk to you about a study that we've had ongoing in the lab for some time now, investigating the temporal characteristics of vowel articulation. And the technique that we use to do this is EMG, or electromyography. So it's pretty well known that articulatory movement typically precedes the onset of an acoustic signal. So just prior to, for example, articulation of a vowel, your tongue muscles, your lips, your jaws all start to move towards this target, and then the voicing begins. Subsequent to voicing, we also have that sort of period of articulator retraction, or moving on to the next position. Okay. Belberti and Harris in 1981 proposed that we call this period of articulator movement the articulatory period, trying to separate out the acoustic signal from the articulator movements. So essentially what they mean by this is a period of muscle activation which encompasses the acoustic signal. So here we have articulator movement starting, the acoustic signal then starts, and then the articulatory period ends over here. Okay. However, there's a lot that's pretty unclear about this articulatory period. So how long is it? How much does it vary? How early is the onset of articulatory movement in advance of the acoustic signal? How late does it stick around? How long does it take those articulators to return to rest position or move on to the next target? And does it vary in predictable ways? So if we're talking about vowel articulation, you would imagine that vowel height or backness should influence when the articulators start to move. You'd also assume that if there were preceding phonemes, that co-articulation should also play a role in influencing when these articulators start to activate. And similarly, you'd think that speaking rate should also influence it. So if you're speaking very slowly, maybe a static vowel, you're just holding a vowel for 2 to 3 seconds, versus if you're speaking at a regular rate, your articulators are probably moving on a very different time scale. And also, too, when we think about all of our articulators, which are made up of multiple muscles, they may have different activation onset and offset times, right? So it may take one muscle longer to activate than another. This is something that's unclear. So essentially what we're trying to do is just provide more information about this articulatory period. And we do that by identifying the onset and offset of muscle activation in relationship to the onset and offset of the acoustic signal. And we do that by looking at exactly these variables in our study. Okay, so first off, how do you measure muscle activation? Ultrasound, real-time MRI, EMA, electropalatography, all of these provide really great ways to look at the temporal basis of articulator movement, but they don't really get at muscle activation. So when we're talking about muscle activation, if you think about what has to happen for a muscle to move, it has to receive a signal from your brain to do so, right? Once it's received that signal, then the muscle fibers can then contract, which results in movement. So if we think about the neural circuitry of how this all happens, here we have the brain stem, and the muscles of interest to us, the lingual musculature, okay? So that first signal to instigate movement originates in the primary motor cortex in the brain. That signal is then sent down to the brainstem to an area called the hypoglossal nucleus. And this is a pool of motor neurons that are responsible for controlling the movements of the tongue specifically. This pool is also very interesting because it receives input from respiratory centers within the brainstem itself. Because lingual musculature, in addition to being used to articulate speech, they also do a lot to maintain your upper airway and keep you free of choking, keep your airflow modulated. Only then does it continue on to the muscle fibers themselves. So when we're talking about dividing this up and trying to figure out how to look at it, people generally take a look at just this sort of area of the circuit, okay? Looking at from the hypoglossal nucleus down to the muscle fibers themselves. And this area, sorry, I'm kind of at an odd angle. This area is essentially that's referred to as a motor unit. So motor units consist of two parts. You have your motor neuron, which is essentially a cell body, which sends that signal. And usually these are located in the brainstem or the spinal cord. So the previous picture I showed you, it was located in the brainstem. That's where the hypoglossal pool is. That's where those lingual muscles are getting their input from. This example in this picture is a motor neuron in a spinal cord. So you see the cross-section of the cord. Okay. And that's right there, the motor neuron. And these are connected by a long fiber to the muscle fibers themselves, and this is called an axon. Okay. And I apologize if any of you are already very familiar with neurophysiology. I just want to get us all on the same page. Okay. So if we blow this up a little bit larger, here we can see the cell body of the motor neuron, and here we see the axon, and out here is where the muscle fibers are going to be. And the length of that axon can vary quite dramatically depending on what it's actually innervating. So if you're thinking about the muscles in your feet being innervated by a motor neuron in your spinal cord, that can be quite a long axon. Okay? So the other part of a motor unit is the muscle fibers that are actually getting innervated. So even though we have these things that can be quite geographically distinct within the body, they're considered one unit. Okay? So electromyography, or just EMG from now on, it's a little easier, allows us to record the commands sent by the motor neurons to the muscle fibers themselves. And when we're talking about these commands, essentially what they are is a series of electrical wave signals or action potentials. So as long as we have an electrode in the muscle fibers themselves, any electrical signal that gets sent into those fibers we can record. We can also amplify it and listen to it, which is pretty cool. So when that signal gets just discharged, it's called firing. And firing rate is essentially the frequency of those action potentials within a train. And a train is just a group of action potentials. So one of the major benefits of EMG is that there is a one-to-one correspondence between the action potential discharged by the motor neuron, which in our case is going to be right there in the brainstem, to the action potential that we record down at the level of the muscle fiber. That's pretty remarkable if you think about it. It essentially gives us a window into the neural activity of the brainstem of a healthy human subject without any casualties. So before going on to the experiments that we did, I just want to give you a brief note about the types of EMG that are out there, because there's a couple kinds. There are whole muscle EMG and single motor unit EMG. So in whole muscle EMG, what you do is record all the electrical activity that you find within a certain region of muscle fiber. So this is signals from multiple motor neurons that are coming down, and it results in a signal that looks like this. Okay? So here you see this big staticky burst, and you can't really pull out individual motor units from this. It's just a whole bunch of input from multiple motor units. In contrast, single motor unit EMG, you record only a single motor unit, and that results in a signal that looks like this. And you can actually you can blow this up and get in even closer, and you can actually see the shape of each action potential. So each of these approaches has different pros and cons. One of the best things about whole muscle EMG is that it provides big picture information about a whole population of motor neurons. So say you're recording from, I don't know, this muscle, for example. It lets you know, is there a lot of activity when you do a certain maneuver? Is there little activity? When does the activity tend to turn on, and when does it tend to turn off? One of the drawbacks to this technique, though, is that within that population of motor neurons, they can act quite differently from one another. Okay? So we don't get any information about, for example, single motor unit firing rate, the variability of the firing, when individual motor units turn on and off. All that sort of stuff is not available in that signal. In contrast, that's exactly what single motor unit EMG is good for. We can record those individual behaviors, and then we do have access to how fast is that particular motor unit sending that signal. Is it variable? Okay, when do individual motor units turn on and off? All of these sorts of, all this level of information. The drawback to this particular technique is that it's very difficult and time-consuming. So if you imagine putting an electrode into muscle fiber, it's full of electrical signals from all of the motor units that are innervating that tissue. Trying to isolate the signal of just one of those takes a lot of time. So in the current study, we have 2 experiments, and we use both of these techniques. Okay. We also have an imaging component, where we did a series of magnetic resonance imaging, and we also did some diffusion tensor imaging. So for both of these studies, we have to think about, well, we're interested in knowing when is the articulatory period, how does the articulatory period vary? We have to select a specific articulator to actually do our recordings from. So if we think about all the articulators that are available, there's a lot: lips, tongue, jaw, velum, etc. And all of these are made up of multiple muscles. Okay? So here are the muscles of the upper airway or the vocal tract. And you can see this is just a sub-sample. There's actually a lot more than this. And they're all active in varying degrees in speech. So for our purposes, what we chose to go with was the genioglossus, and that's right there. Okay? So this is a fan-shaped muscle. It essentially is composed of horizontal fibers, which start right there, and they fan out into oblique fibers, all of which would insert right into the tongue body there. And this muscle originates from the mandible bone. Okay? So there is a skeletal insertion. We chose this muscle specifically because it's the principal protruder muscle of the tongue. It's also one of the biggest lingual muscles, and it's the major mover of the tongue body itself. So as such, you'd think it'd be pretty active during speech. And it's also, compared to a lot of these other muscles, fairly easy to access in terms of intramuscular EMG. Okay, so when you're thinking about EMG and what it can tell you about muscle structure and muscle function, you have to first understand what structurally the muscle you're recording from is actually capable of doing, okay? Because oftentimes form equals function in this realm. So the tongue is part of a unique category called a hydrostat. I'm sure many of you are familiar with this concept. The many other structures in nature are also hydrostats. So for instance, slugs, octopus tentacles, things like this, okay? So a hydrostat is a structure of fixed volume. What this means is if you think about, I don't know, like a bag of goo, essentially, if you have compression in one area, this results in displacement in another. But the overall volume is fixed, okay? There's no skeletal support for a hydrostat. So what that means is that other muscles that it inserts into, because instead of inserting into bone, it inserts into other muscles. When these muscles contract, that's what forms the skeletal support for that muscle to maintain force against. Because of this, the tongue has a very high number of degrees of freedom. So to get from point A to point B, there's a lot of options, basically. The GG is a major offerer of that structural support. Okay, so contractions of this muscle basically allow the tongue body to move against it. So before we did our EMG study, we did this MRI study to have a better sense of what structurally contractions in the GG, how that results in tongue displacement in the rest of that area. So for this study, we had 4 subjects, 2 male, 2 female. And we just had them lay in the MRI tube for 2.5 minutes and hold vowel articulations. Okay, it's actually quite difficult. I felt bad for them. Let's see. So, as I mentioned, the GG consists of 2 fiber orientations, which are said to have 2 distinct functions. So here's an MRI image of the tongue at rest. This is a sagittal view. The genioglossus is located right there. Okay. Here you can see the attachment at the mandible, and you can see how it inserts into the tongue body. Now when we take the same look at that sagittal view of the tongue articulating E, note the difference. Okay? So here you can see we have a contraction in those horizontal fibers of the GG, which compresses the tongue root or tongue base, which results in displacement up here, resulting in that height of that high front vowel. If you compare that to the rest condition, you can see it's quite different. When we take a look at a low back vowel, and I apologize, this is a same subject, different day, so the field of view is a little different, but for our purposes it'll work. We can see that the contraction is no longer in the horizontal fibers, but instead in those oblique fibers. Notice the displacement points are on either side of that. Is a low back vowel. It's relatively low in the oral cavity, and you can see the displacement back near the base. If you take a look at the pharyngeal width differences, they're pretty marked. So essentially what this tells us is that posterior horizontal fibers draw the tongue root forward and displace the volume of the tongue body upward. And this is something that had been proposed before. This is not a new revelation. Honda in 1996 has done a lot of work on this. In contrast, the anterior oblique fibers compress and retract the tongue body. Okay? And there's also a lot of evidence to suggest that these two different regions of the GG are controlled in two different ways. There's suggestion that they have different innervation points, all from the hypoglossal, but that they have separate motor end plates, for example. Okay, so we wanted to know more about those muscle fiber orientations, not only to get a sense of direction of contraction possibilities, but also to know where exactly are we putting our electrodes and what is likely to occur at those locations. So we did a kind of imaging called DTI, and some of you may have seen this before. I included this picture I got from the internet. They do this a lot with brains, and it shows the neural tracks in the brain. Okay. Essentially how this works is it's three-dimensional imaging, which is really nice for our purposes because it results in an image that we can rotate in three-dimensional space and really investigate. It works by identifying the orientation of water molecules in muscle fiber, and this, helpfully for us, matches the orientation of muscle fiber fascicles. So once we have this image, we can then process it using probabilistic tractography, which creates a macroscopic representation of muscle fiber bundle orientations. So that's a fancy way of saying it can make a pretty good guess based on the sort of criterion that you give it, where those muscle fibers are going to be based on the information on the water diffusion directions. For this study, we just had one subject. We scanned from 30 different directions, and because this particular imaging is very time-consuming, it took 14 minutes of him lying absolutely still, we had him lay with the tongue at rest. Okay, so this is pretty cool. We were excited about this. This hasn't really been used a whole lot for lingual musculature. I think maybe once before in humans and another time in a cow tongue. So this is one of the images that we got. And here you can see most of the lingual musculature. Right down here we have the geniohyoid. Right here we have the hyoglossus. Behind it is the genioglossus. Okay? This out here is probably part of the styloglossus. And you can see very nicely the insertion into the mandible and the extension into the tongue body proper. This is another view of it from behind slightly. And here you can see even more clearly what these upper fibers of the GG, how they extend right up. And if we had continued processing this image, the tongue body would lay on top of it. And it inserts right into those fibers of the intrinsic tongue muscles, which essentially run like this. Okay? Here is more pared down. You can see a little bit more of the genioglossus, and here the genioglossus is isolated. Okay, so this is our muscle that we're going to be recording from. This is an inferior view underneath the chin. It's important to remember that this muscle has two bellies. Okay, it's double-sided, right? So underneath there are two sides. In the middle there's a strip of basically fascia or stiff tissue that divides them. And this is a view from up above. So before it starts to insert, it's a very nice view of those oblique fibers, and you can really see that clear angle of the fibers. Okay, that was a little bit gratuitous. We just really like those pictures. Okay. All right, so moving on to our first experiment. We had 8 subjects, 4 male, 4 female, all undergrads here at the University of Arizona. Our hypothesis for this experiment, simply that degree of displacement for the tongue from rest will alter duration of EMG activity. Okay, specifically what this means is that we think when the tongue moves forward or upward, it should yield longer EMG activity durations. We also hypothesize that speech rate will influence this, as well as muscle regions. So remember, we know that those 2 different muscle regions have 2 different functions. So we should expect some sort of interaction between vowel and muscle region, depending on whether it's a high, low, or front-back vowel. To do this, we had subjects produce vowels in 3 different contexts while we recorded audio and EMG activity in the GG. Those vowel conditions were, first of all, static vowels. So we just had subjects produce vowels in isolation, e, u, a, and ah, and they held them for roughly 3 to 4 seconds each. Okay. We selected these particular vowels because they occupy the four corners of the vowel space. So they give us really nice permutations of tongue height and front backness. And here we see some more MRI images that we created, I, U, A, A, and you can see this should give us a wide variety of GG action. We were also interested in co-articulation with a preceding consonant. So we also included a fricative plus static vowel condition. So here we selected two different fricatives, the coronal S and the palatal esh. Again, these were static vowels, so they just sort of held it for 2 to 3 seconds. We selected these two fricatives specifically for their differences in the tongue body position. So S, as you know, the tongue tip is positioned forward on the palatal ridge, but for esh, the tongue tip and body, or blade, approximate the hard palate with a midline groove. So these are two very different tongue postures, and we were curious to see what does the addition of this consonant onto that static vowel do to what we found in the first condition. And then because both of these conditions involve static vowels, we wanted something that approximates real speech a little bit more closely. So we had a vowel embedded in a nonce item. And because we could, we did 11 vowels. Okay, so we have things like a peep, a poop, a pap, a pop, etc. Okay. So in terms of our EMG recording and apparatus, before we do any kind of insertion with an electrode, first we do an ultrasound of each subject to identify the depth and location of genioglossus. It varies a little bit between people, body mass, height, all of these things influence the exact depth for people, so we know exactly how far in to put the electrode. Let's see. Oh, it also rules out whether they have any vasculature down here. If they do, they can't participate. So our two recording sites are the anterior GG, which is those horizontal fibers right there, or excuse me, the oblique fibers right there, and the posterior GG, so right down here. Okay. So to insert the electrode in the anterior GG, we used two bared bipolar hook wire electrodes, and they were inserted using a hypodermic syringe per orally into the floor of mouth. So what that means, it sounds awful, it's really not, I swear. So what that means is you have a very, very, very fine wire, essentially as thin as one of my hair strands, okay? That's threaded through a needle, and using the hypodermic syringe, you just pop it in, twist the needle out, and the hooked wire is left embedded in the muscle fiber, and you can't even feel it, okay? That's inserted right about there, okay? Some of you have done it, so you could potentially attest that we're not making people cry. Depth of insertion is 1 to 2 centimeters. In the posterior GG, we have to use a different kind of electrode and a different type of insertion technique. We can't have syringes in this area. It's just not good. So for this particular area, we use 2 tungsten microelectrodes. And that essentially just looks like a needle, okay? But it's a little bit thinner. It's about like an acupuncture needle. So these were inserted percutaneously, so they go right there through the chin, okay? And these have to go a little bit away from midline because we want to make sure we're in the middle of one of the bellies, not in that fascia, and about 1 to 3 centimeters posterior from the mandible, so basically right in the middle. And depth of insertion here is usually a little bit deeper than in the oral cavity because we have to bypass the other muscle underneath the GG right there. And it's usually 1.5 to 3 centimeters, again, depending on the subject. So, to measure our EMG activity onset and offset, we developed 2 dependent variables that we're just calling firing onset time, FOT, and firing offset time, FOFT. This is an example of a record we might generate. So here we see across the bottom, the subject's articulating C with the S plus the E static vowel, and they're just doing serial articulations. Here we have the anterior GG activity and the posterior GG activity. Notice how different they look. So if we see the anterior, you can see it's kind of lighting up for the bulk of that vowel, but interestingly, here in the posterior, it's just really lighting up for that coronal S, which is quite interesting. Yes?
And then that's 1 second there at the bottom?
Correct. That's the scale. So it's pretty fast. It's pretty much instantaneous. In order to analyze this, we generate what are known as rectified integrated traces, which essentially change this activity into a wave where departures from 0 equal activity. It's just easier to analyze. So to calculate FOT and F off T, we noted the onset and offset of the acoustic signal, A and B, and then we noted the onset and offset of the EMG activity, X and Y, and to get FOT, we simply subtracted the onset of EMG activity from the onset of the acoustic signal, and vice versa for F off T on the other side. To analyze, we used a linear mixed effects regression model fit to the following conditions. Random factor with subjects, because here we have no motor units to call random. There's just a whole signal. Our fixed factors were vowel, muscle region, anterior versus posterior, and place of articulation of preceding fricative, whether it was coronal or palatal. Our results. So EMG durations were longer than utterance durations regardless of task. So essentially, no matter what subjects were doing, here we have their utterance duration in blue, the activity that was generated in the genioglossus was always longer. Okay, so that's good. The averages for FOT and F off T were always ended up being positive, which tells you that for the most part, activity started before the onset of the acoustic signal and ended after the offset. There were some cases where it started after the onset or ended before the offset, and that happens, but the majority of the time, it's before and after. So let's just take this condition by condition, starting with static vowels. For activity onset, we found a significant main effect of vowel. And post hoc contrast revealed that the front vowel activity preceded back vowel activity by approximately 100 milliseconds. And here I laid out all of the vowels according to muscle region, just so that you can see what was happening rather than just giving you 2 bars, because it's not that interesting. And you can see across the board how this varied. We did not find any significant effects of muscle region for the onset of activity for this. So it didn't really seem to matter whether you were anterior or posterior. The front vowel activity preceded the back vowel activity. In terms of offset, this is where we found a main effect of muscle region. So here we found that the anterior activity tended to turn off 170 milliseconds before the posterior. Okay, so we see particularly it's marked in those back vowels right there, how long that posterior activity really went on for. Moving on to the fricatives plus static vowels. Here we found a significant interaction of vowel and place of articulation of fricative. Specifically, we found that in the front vowels, sa preceded c by approximately 126 milliseconds. And in the front vowel a, the coronal, sa, preceded sha by approximately 115 milliseconds. Again. We did not find any significant effects for activity offset in this particular condition, which is interesting considering that we did for the previous. And you can notice, too, that the duration of the offsets are much shorter in this condition than they were for the static vowels. Moving on to the nonce word condition, we found a significant main effect of muscle region here. And I've arranged the vowels here so that they are on an axis of height, starting with the high vowels over here and ending with the low here. And post hoc contrast revealed that the posterior activated approximately 80 milliseconds before the anterior. And you can see that how marked that is within that high vowel range. Those green bars are really sticking right up. For offset, we found a significant main effect of vowel and muscle region. And specifically, we found that for both the high and the low vowels, the anterior turned off 80 milliseconds before the posterior. So again, we see that long duration in the posterior muscle, region of the muscle. So these findings support our hypothesis, simply that degree of displacement from rest will alter the duration of EMG activity. Specifically, that movement of the tongue forward or upward should yield longer EMG activity durations, and that vowel height and front backness alter the onset and offset of this articulatory period. However, we found that this effect differed between speech task or speaking rate, and that it was different between regions of the same muscle. So if we're talking about just the static vowel, we find overwhelmingly that activation begins significantly earlier for front versus back vowels. In the fricative plus static vowel condition, we found, again, activation begins significantly earlier for the coronal versus palatal consonant. So all of this sort of combined suggests that forward movement of the tongue results in earlier activation times regardless of muscle region. But when we look at the non-sitem vowels, which are the most temporally similar to real-time speech, we find activations between regions of the GG differ. We found that posterior GG activity begins earlier and lasts longer than the anterior GG, and that we found particularly marked effects for high and low, but not the mid-vowels. So all of this seems to suggest that vertical movement of the tongue in this condition results in earlier onset and later offset of the articulatory period within the posterior, specifically GG. So these differences between GG regions for high and low vowels point to different roles for these regions, which we had kind of anticipated. The posterior GG, as I said, the horizontal fibers draw the tongue root forward and displace the tongue volume upwards to create that height. So articulation of high vowels necessarily entails activation of posterior GG. Yet the activation lasts longer in posterior GG for not only high vowels, but also low vowels. So this suggests that not only is posterior GG providing support to articulation of high vowels, but that it also works to sustain low vowel articulation. And as I mentioned before, previous research has proposed that hydrostats rely on muscle contractions to provide that skeletal support frame in order to maintain force. So potentially, in this low vowel condition, the GG is providing that sort of auxiliary support to maintain that depression. So moving on to experiment 2. We did experiment 2 largely to confirm that the whole population recordings we did in experiment 1 were also found to be consistent on a single motor unit level. Because as I mentioned, single motor units within a population can really do quite different things. We wanted to see, do we see these same effects? So for this particular experiment, we recorded single motor units. It allows for examination of motor units within a larger population. We examine those same temporal characteristics, onset and offset of activity, but then with this particular technique, we can also add in some new variables, specifically firing rate, so how fast those action potentials are getting fired. And firing rate is a measure of motor unit activity level or contraction force. So if we think about if the motor unit is firing away at a really, really high speed, this suggests that there's an increased activity level. This means there's an increased excitation and drive to that motor neuronal pool that's associated with those fibers. Our hypothesis for this, in line with the findings of the static vowel condition in the previous experiment, simply we think front vowels, we expect them to yield earlier FOTs and later FOFTs. For firing rates, we really weren't sure what to predict here, so we didn't. Our 2 variables were mean firing rate per utterance and standard deviation of firing rate per utterance, so the variability of the drive. Okay? We don't have any directional hypotheses. We just weren't even sure what would happen. For this particular experiment, we had 31 healthy adult participants. Our EMG recording, we had single motor units were recorded from the posterior GG only. It's difficult. For this particular type of EMG, you have to use a tungsten microelectrode, and you can't put a hard needle into someone's mouth and expect them to speak. It's not going to work. So tungsten microelectrodes were inserted percutaneously in the same fashion as before. In this protocol, we had subjects produce static vowels, U, and ah, while we simultaneously recorded audio and EMG. Note that there's no Nonce condition here. There also is no fricative plus static vowel condition. And the reason why is because I mentioned before, SMU recording is very difficult. The subject has to maintain, their head has to be supported, they have to maintain a position of extreme stillness. Their jaw has to remain open for these serial articulations. And we insert the electrode, and you can imagine all of the electrical activity. We're trying to isolate just the signal of one motor unit, and it's done by tiny little adjustments of this electrode. As soon as we find one, we immediately hit record. Everybody holds their breath, basically, and we try and get a complete recording done. This is an example of data that we would generate. So here across the bottom, we have the single motor unit trace, okay? And down here, this is one of these are called spikes. These action potentials and spikes are the same thing. This is one of the spikes blown up large. Interesting things motor units is that their action potentials have a characteristic shape. It's one way we know whether it's the same motor unit or not. So here, these are all these spikes from this trace overlaid on top of one another, and you can see that they match one another in shape. So this tells us it's the same motor unit. In addition, the timing and also the same amplitude tells us it's the same motor unit. Okay. In the second trace, we have the instantaneous firing frequency. So here along this axis, we have hertz. So how fast is this motor unit firing? Here we have the acoustic signal, and in this particular example, it's the vowel ah. So our statistical analysis, we again used a mixed effects model. This time, our random variables were subject and motor unit. Our dependents, or fixed, were FOT and F off T, and the mean firing rate per utterance and the standard deviation of firing rate per utterance. So in order to generate these last two, we take the mean firing rate just within this boundary of the utterance onset and offset, and we also take the standard deviation within this too. We recorded a total of 111 single motor units, which is a lot. If you read a lot of the previous work on this, most people top out at 8. So we decided just to go all the way. The articulatory period in this condition, just like the previous experiment, utterance duration was typically encompassed by the firing duration. The results, we found a significant effect of vowel. Again, we found that front vowel activity turns on significantly earlier than back vowel. So good, we're consistent. We also found a significant effect of standard deviation of firing rate. So such that as the FOT decreased, so as the articulatory movement got closer and closer to the onset of the acoustic signal, the standard deviation, the variability of that firing, increased. We found no other significant effects on FOT, so average firing rate didn't do anything. For F off T, we found no significant effects as well. So, fortunately, our SMU results support our hypothesis. They're consistent with the findings in the previous static vowel condition. Front vowels yielded significantly earlier FOT. The F off T's did not match the whole muscle static vowel condition, and potentially this is maybe a function of shorter activity durations in general. We don't know. It's something for future research. In terms of our firing rate variables, as FOTs got shorter, the variability of activity increased. This suggests that tongue control is more stable with more onset time. So for whatever reason, if you have more time to prepare for that gesture, that firing rate is a little bit more stable. Potentially, you could interpret this to mean that maybe tongue movements are more precise with more onset time. So when we compare our conditions, in the static vowel conditions, again, we found that forward movement of the tongue instigated earlier onset and later offset of activity, regardless of GG region. Didn't matter. And this was supported by both whole muscle and single motor unit data. In the nonce item vowel condition, we found that vertical movement of the tongue instigated earlier onset and later offset of activity, specifically in the posterior GG. So based on these two findings, we can conclude, obviously, speaking rate is an important variable when trying to identify influences on the articulatory period. In terms of implications for linguistic research and what this might all help us to figure out, differences in effects for static versus nonce item vowels strongly suggest that static vowel articulation is subject to a different kind of speech motor planning than, for example, casual or real-time speech. And potentially, you could also infer that precision of tongue movements may vary as a function of firing rate variability. So when we talk about target undershoot or overshoot, this might be another way to think about this. When we collect speech samples for phonetic analysis, of course, this is something to bear in mind. What exactly is it that we are analyzing when we collect, for example, careful versus not careful speech? In terms of the effects of tongue height, front backness, and differences between muscle regions on the articulatory period, these bring new considerations, for example, on the use of response latencies as a reliable measure in perceptual and psycholinguistic tasks. So response latencies, the assumption behind them is that the amount of time it takes to initiate an oral response is the same, regardless of phonetic content. And we use these latencies to tell us something about cognitive organization, and they're incredibly helpful. However, the current findings suggest that response latencies are subject to the influence of all of these variables. Okay? Mechanistic considerations are clearly a part of it. How long it takes the tongue to get from point A to point B may differ depending on these variables. And then our interpretation of response latencies might need to be more cautious, and potentially we should be balancing our items for at least phonetic content. So these findings underscore the complexity of timing of articulator movements. We all knew it was very complicated in there. This upholds that. More insight into physiological mechanisms underlying speech are clearly needed. We're just barely scratching the surface on this sort of research. In particular, the motor control of lingual musculature in speech is extremely under-researched. These muscles have been very well, the motor control of these muscles in terms of their role in respiration has been very, very well studied, but the physiologists are not thinking about speech. They're not thinking about, oh, we use these muscles to articulate language. There's a funny story about one of the physiology faculty. He was asked by somebody from NIH what role the tongue had in speech. He couldn't answer the question. So this is an area in which we have a great deal of crossover to offer. On the converse side, I think that it's helpful to think about the multiple role of these muscles, not only in speech but also in respiration. These muscles are used to modulate an airflow. So essentially, we need better understanding of neural control of articulators. We need to understand how this control is mediated by other drives, such as respiratory. And we need to understand better how the mechanistic dynamics of articulators interact to achieve speech targets. So if we do all of this, potentially accurate understanding of speech gestures and their timing relations can provide more realistic basis to theories about speech production and specifically speech planning. Thank you. This is one of our lab people. He has an electrode in right now.
Which electrode is that?
This is the tungsten. It goes in underneath the chin right here.
In the static condition, or any of the conditions where your subjects are producing the same thing multiple times,